Alrighty, ladies and gents, we're going to keep on keeping on with Chapter 8. We've now covered sort of an introduction to cell division. We've talked about mitosis in eukaryotic cells. We are now going to talk about meiosis in eukaryotic cells. So they're related but different processes. So meiosis is used to make what we call sex cells. The rest of your body's cells, or the rest of the body cells in all of our different living species, are what we call somatic cells. And they contain a very specific number of chromosomes, which is species specific. So for instance, human cells have 46 chromosomes, so 23 pairs, what we call homologous chromosomes. Chromosomes of a homologous pair basically carry the gene for the same characteristic at the same place, and we call that a locus on the chromosomes. So you get one set of chromosomes from your mom, one set of chromosomes together from your dad. Those are homologous chromosomes. So you can see here we have our sister chromatids, right? And here you have a duplicated pair of homologous chromosomes. And you can see each of these, the colors are used to represent sort of a specific locus on the chromosome, a specific spot where the chromosomes are going to carry information for a specific trait, a specific gene. Now the number of chromosomes that a species does differ. For instance, the cat who's trying to climb up on my lap here has a different set of chromosomes than I do, but the same setup. 23 pairs, one from the male, one from the female. We've got our homologous chromosomes. So the cells, the body cells, the somatic cells with the two sets of homologous chromosomes are considered what we call diploid. But our sex cells, our gametes, eggs and sperm, they are considered haploid cells. They have a single set of chromosomes. Of course, if they had a full set of chromosomes, if we didn't split the chromosomes and split them, reduce that chromosome number, essentially we'd be doubling the number of chromosomes every time we go through a round of reproduction. And so in animals that produce, or in organisms that produce sexually, you sort of have this alternation of haploid and diploid stages. Let me show you what I'm talking about. So here, right, you've got these multicellular diploid adults. In their sex organs, they're going to make the gametes. Those gametes are haploid, so they have half the number of chromosomes. That's the eggs and the sperm. The sperm fertilizes the egg. Then we have a diploid zygote. That zygote goes through mitosis and development, ends up a multicellular adult, which produces the sex cells, and so on and so forth. So this sort of alternating haploid-diploid. Because we are reducing the number of chromosomes, each of these cells is going to go through not one round of division, but two. So here is our pair of homologous chromosomes. Again, one from mom, one from dad. They're going to duplicate, and so they each have then a sister chromatid. The homologous chromosomes separate, and then the sister chromatids separate. And so the result is not 2 daughter cells, but 4 daughter cells that are haploid, not diploid. So we're going to go through one round of multiplication, duplicating the DNA and the chromosomes, but 2 rounds of cellular division, meiosis 1 and meiosis 2. So there are similarities between mitosis and meiosis, but then there are differences, right? Like mitosis, before a cell goes through meiosis, the chromosomes have to be duplicated. During meiosis 1, the homologous chromosomes will sort of pair up into something called a tetrad. And what happens is this really interesting thing called crossing over, where the homologous chromosomes basically sort of pass things back and forth between each other. The homologous pair separates. That results in 2 daughter cells, each with 1 set of chromosomes, still joined together as those sister chromatids. Then the sister chromatids then separate during meiosis 2, and we get our 4 haploid daughter cells. So here during interphase, the chromosomes duplicate. Again, the DNA is in that chromatin format. The phases of meiosis 1 and meiosis 2 are very similar to the phases of mitosis: prophase, metaphase, anaphase, telophase. But since we are here in meiosis 1, we're talking about prophase 1, metaphase 1, anaphase 1, etc. So here you can see the sister chromatids. You can see the formation of the mitotic spindle. You can see the nuclear envelope has disintegrated. And the homologous chromosomes have kind of matched up together into these 4 pairs, what we call a tetrad. And then during metaphase 1, as the chromosomes sort of line up on that imaginary middle metaphase plate, this is when this sort of crossing over process happens. They sort of swap DNA. That results in sort of genetic variation now in the sister chromatids. Now, the chromatids are still attached to each other, but the homologous pairs will be separated, pulled apart during anaphase 1 as the cell elongates. We will get cytokinesis and telophase 1, where the mitotic spindle will sort of disintegrate. Then from there, we'll get the formation of another mitotic spindle and move into meiosis 2, wherein the sister chromatids will separate. So again, they sort of line up on that metaphase plate during metaphase 2. The sister chromatids then are pulled apart during anaphase 2. And then during telophase 2, looks very similar to the last phase of telophase in mitosis. The chromosomes start to uncoil, the nuclear envelope starts to form again, and you get cytokinesis, resulting in 4 daughter cells that are genetically distinct from the parent cell and genetically distinct from each other. So if you were to compare mitosis and meiosis, it's still just a single duplication during the S phase of interphase. During prophase, the duplicate chromosomes remain separate. During metaphase, those sister chromatids line up at the metaphase plate. And then during anaphase and telophase, the sister chromatids separate. You get cytokinesis, 2 genetically identical diploid cells. So this is what goes on during our growth processes, whenever we do sort of tissue repair, and what occurs in asexual reproduction. Conversely, so this is our somatic cells, our body cells. For just the sex cells, they're going to go through meiosis. So during meiosis 1, the homologous chromosomes come together. That's where we're going to get some crossing over. They sort of swap. The pairs of the homologous chromosomes line up during that metaphase 1 sort of imaginary middle plane. They split into 2 genetically distinct daughter cells. The chromatids separate, and then we have 4 genetically unique haploid cells. So meiosis is only used in sexual reproduction. No organism that reproduces asexually uses meiosis. And if we're talking about like making new skin cells because you shed skin cells every day, that is a mitosis process, not a meiosis process. And if you're having a hard time visualizing this, I've got some videos to show you guys. particularly this crossing over a bit, because I think that sort of confuses people. Right, I have 4 siblings. We all look very different from each other. We all act very differently from each other, right? There's genetic variation. If you have siblings, you know that, right? Sometimes your siblings are very different from you. And so we get this sort of, it's called independent assortment, right? Meaning the chromosomes orient themselves very independently. You get random fertilization. The chromosomes at each in each homologous pair sort of differ, sort of in many different places, and sort of how they kind of line up during the earlier processes in meiosis leads to like a whole many combinations, tens and tens of trillions of combinations of ways that sperm and egg can combine to each other. So we get sort of random fertilization. And then we have this crossing over process, this switch of genetic material, which then greatly even further enhances the amount of variation and variability that you can see in the offspring of sexual organisms. So here, for instance, here is 2 equally probable arrangements of the chromosomes at metaphase 1. When the chromosomes line up on metaphase 2, you can see this is one possibility, this is one possibility, but this is just as equally possible, and this is just as equally possible. They're all as equally possible. And then when they split again, you can see we're sort of getting this greatly enhanced variation, right? All 4 of these combinations are as equally possible. And that's just one round of meiosis. And literally you have billions of sperm that are being made every day. So there's just a huge variety in terms of how the chromosomes line up and how they sort. So this sort of independent random assortment introduces variation to a very large degree, literally trillions and trillions of possibilities. But then you have this other sort of inherent variation that occurs due to the crossing over. So there do exist differences between the homologous chromosomes. Even though they're carrying information for the same trait, they may not be carrying sort of the same version of that trait. Here you can see at this locus, we have the genes for the color of the rat's coat, and here we have the loci for the eye color for these rats. And they're going to independently assort, and the resultant offspring can have sort of any combination here. They could have a brown coat and black eyes, they could have white coat and pink eyes, or some combination therein. Because what happens during this crossing over process is that the non-sister chromatids, so not the ones that contain the same identical DNA, but the sort of the red pair and the blue pair, right? The homologous chromosomes sort of swap. And so you can end up with more variation that way. So you can see here are some non-sister chromatids, and you can see they're sort of swapping some information there, right? But you can get a little bit of the blue gene to the red. If we're looking at this just in color, right? A little bit of the blue comes over to the red, a little red goes over to the blue. That little crossover point is called a chiasma. Chiasma just means to cross over. And that results in just this huge amount of variation. So here you can see the paternal chromosomes. Here you can see the maternal chromosomes. They're in red. When they line up there during metaphase, the non-sister chromatids, they form those chiasma. They cross over. So you're getting sort of hybrid chromosomes. Here's a mostly maternal chromosome with some paternal DNA. Here's a paternal chromosome with mostly paternal chromosome with some maternal DNA. And then when the homologous chromosomes separate, and then when the sister chromatids separate during meiosis II, you get some of those cells that may look very similar to the parents, but some are going to have that variation in them and that genetic variability, which is why some traits that you have are very similar to your parents, some traits that you have are different. Siblings can all be different from each other, so on and so forth. It's this chiasmata, this crossing over, that greatly enhances the genetic variability that we see in sexual reproduction.